A paper published today from MIT claims that the enzyme you are using right now to breathe oxygen is at least 2.8 billion years old. That would mean life on Earth figured out how to consume oxygen hundreds of millions of years before oxygen ever became a permanent part of our atmosphere. And if that is true, it doesn't just rewrite the history of breathing. It might explain one of the biggest unsolved mysteries in Earth science. And it has some uncomfortable implications for how we search for alien life. Okay, so here is the setup. You probably know that early Earth had basically no oxygen. The air was mostly nitrogen, carbon dioxide, methane, toxic to us, completely unbreathable. Then, around 2.3 billion years ago, something massive happened. Oxygen levels in the atmosphere jumped. Permanently. Scientists call this the Great Oxidation Event. It is arguably the single most important chemical transformation this planet has ever gone through. Without it, no complex life, no animals, no plants, no us. Now, the organisms responsible for making that oxygen were cyanobacteria, tiny photosynthetic microbes. They figured out how to split water using sunlight and release oxygen as a waste product. And here is what has been bugging scientists for decades. We have pretty strong evidence that cyanobacteria evolved the ability to produce oxygen around 2.9 billion years ago. But the Great Oxidation Event did not happen until 2.33 billion years ago. That is a gap of roughly 600 million years. 600 million years where oxygen was being produced but was not accumulating. To put that number in perspective, 600 million years is longer than the entire age of animals on Earth. Everything from the Cambrian explosion to the dinosaurs to right now fits inside that gap. So what was happening during all that time? Where was the oxygen going? The traditional answer has always been geology. Rocks ate it, iron dissolved in the ancient oceans, reacted with oxygen to form iron oxides, which sank to the bottom. That is actually where most of the world's commercial iron ore comes from, by the way. Those red banded iron formations you see in geology textbooks. Those are basically the graveyard of early oxygen. Volcanic gases consume more of it. Reduced minerals on land absorbed even more. So the standard story was that geological sinks kept pace with biological production for hundreds of millions of years, until eventually the cyanobacteria just overwhelmed the system and oxygen broke through. That story makes sense. But it has always felt a little incomplete, because the numbers are hard to balance. 600 million years is a really long time for rocks alone to perfectly cancel out a living, growing, evolving source of oxygen. Something else might have been helping. And that is exactly what this new MIT paper proposes. The team, led by Fatima Husain and Gregory Fournier, focused on a specific family of enzymes called heme-copper oxygen reductases. If you have never heard of them, you have definitely used them. These enzymes sit at the end of the aerobic respiration chain. They are the molecular machinery that grabs oxygen and converts it into water, generating energy in the process. In your body right now, this is happening inside your mitochondria. Complex 4 of the electron transport chain, same enzyme family. More than 90% of all oxygen consumed by living things on Earth passes through these enzymes. What the MIT team did was trace the evolutionary history of this enzyme family using molecular clock analysis. They gathered nearly 36,000 genetic sequences from organisms across all domains of life: bacteria, archaea, eukaryotes. They filtered that down, built phylogenetic trees, and used fossil calibration points to estimate when different branches of this enzyme family first appeared. Their result: the enzyme family dates back to the Mesoarchean era, somewhere between 3.2 and 2.8 billion years ago. Even their most conservative estimate, where they removed a couple of deeply branching sequences that could skew the dating, still lands at around 2.9 billion years ago, before the Great Oxidation Event, potentially right around the same time cyanobacteria were just getting started with oxygen production. Think about what that means. Almost as soon as some microbes started making oxygen, other microbes evolved the ability to consume it. Not millions of years later, not after oxygen had a chance to build up, almost immediately in geological terms. Now imagine the scene. And this is not just speculation. We have a modern analogy for this. Today, in places like Shark Bay in Australia, you can find living microbial mats, Layered communities of microorganisms. Scientists have stuck tiny oxygen sensors into these mats, millimeter by millimeter, and measured the oxygen levels at different depths. What they find is striking. Right at the surface, where cyanobacteria are doing their thing, oxygen levels spike, but go just a few millimeters deeper, and oxygen drops to zero, completely anoxic. Other microbes in the mat are consuming the oxygen faster than it can diffuse downward. Now rewind 3 billion years. Picture a shallow, sun-drenched coastline on the early Earth, a thin microbial mat clinging to the rocks. At the very top layer, cyanobacteria are pumping out oxygen in the sunlight. But right next to them, within millimeters, other organisms are grabbing that oxygen and using it for energy before it ever has a chance to escape into the atmosphere. Oxygen exists, but only as a local commodity, a molecule produced and consumed in the same tiny neighborhood. The atmosphere above, still essentially oxygen-free. This is what scientists call an oxygen oasis, and it resolves the apparent contradiction in the title of this video. Life can use oxygen without the planet having oxygen, because oxygen does not have to be a global atmospheric gas to be biologically available. It just has to exist locally, even at trace levels. And lab studies have shown that some of these enzymes, particularly the high-affinity types, can function at oxygen concentrations as low as 1 nanomolar per liter. That is essentially nothing, a few molecules here and there. So what the MIT paper is really proposing is a biological break on oxygenation. Not just rocks consuming oxygen. Life itself was a sink. Cyanobacteria were the faucet. Rocks and oxygen-breathing microbes were the drain. And for 600 million years, the drain kept pace with the faucet. Only when oxygen production finally overwhelmed all the sinks, geological and biological, did the atmosphere tip into a new state. It is a tug of war. And biology was on both sides of the rope. Now, I want to be honest about the limitations here, because this is not a settled case. Molecular clock dating at these timescales, we are talking billions of years, is genuinely difficult. And there are real reasons to be cautious. First, the deeper you go in time, the more your estimates depend on model assumptions. Which mathematical model you use for how mutation rates change over time, where you root the evolutionary tree, how you handle the fact that bacteria do not leave fossils, so your calibration points are indirect. The MIT team tested multiple clock models and 2 different tree topologies, which is good practice. But this is still a field where changing a few inputs can shift your dates by hundreds of millions of years. In fact, in their own analysis, removing just 2 deeply branching archaeal sequences moved the estimated origin of one enzyme group by about 300 million years. That is not a small wiggle. That is the difference between this story being revolutionary and this story being interesting, but less dramatic. Second, there is a directly contradictory paper, published just a couple months earlier in December 2025, by a team led by William Martin at Heinrich Heine University in Düsseldorf. That group argues the opposite, that oxygen reductases, including the ones the MIT team studied, all arose after the Great Oxidation Event, not before it. Their model puts the origin of these enzymes at around 2.1 to 2 billion years ago. The gap between the 2 papers is enormous. We are talking 700 million to over 1 billion years of disagreement. The Martin team emphasizes horizontal gene transfer as a confounding factor. These metabolic genes get passed between unrelated organisms all the time. When that happens, the gene tree does not match the species tree, and your dating can get thrown off. The MIT team accounts for this. They actually use horizontal gene transfer events as additional calibration constraints, which is a technique the Fournier lab has developed. But the fundamental tension is real. Different methods, different assumptions, different answers. So where does that leave us? Probably somewhere in the middle of a genuine scientific debate that is going to play out over the next few years. The MIT paper is well constructed, it is peer-reviewed, the data and methods are published for anyone to check. But it is not the final word. What I find most compelling, though, is that even if you take the conservative end of their estimates, the oxygen-consuming enzymes still predate the Great Oxidation Event. The debate is really about how far before. Was it 300 million years before or 800 million years before? Either way, the basic insight holds. Life was breathing oxygen before the sky was full of it, And there is one more angle to this that I think is the most profound, and it has nothing to do with Earth's past. It is about the future of searching for life on other planets. Oxygen has always been considered one of the strongest biosignatures we could detect in an exoplanet's atmosphere. The logic is simple. Oxygen is highly reactive. It does not stick around on its own. If you see it in a planet's atmosphere, something has to be actively producing it. And the most likely something is biology. This is a huge part of the scientific justification for missions like NASA's planned Habitable Worlds Observatory, which is designed specifically to look for oxygen and ozone in the atmospheres of Earth-like planets around other stars. But Earth's own history shows a devastating problem with that logic. For the first 2+ billion years of our planet's existence, there was life here, photosynthetic life even, producing oxygen. And according to this new study, there was also life consuming that oxygen. An alien astronomer pointing their telescope at Earth during those 2 billion years would have seen no oxygen in our atmosphere. They would have declared this planet lifeless. A study from 2017 estimated that for most of Earth's 4.5 billion year history, atmospheric oxygen was below 1/10 millionth of current levels. That is far below anything we could ever detect from light years away. Earth was a living planet hiding its own biosignature for longer than it has been showing it. Now add the MIT finding. It is not just that rocks were scrubbing the atmosphere clean. Life was actively participating in its own concealment. Organisms eating the oxygen before it could escape. If this happened here, on Earth, there is no reason to think it could not happen on other planets too. A world could have a thriving biosphere, photosynthetic organisms pumping out oxygen, other organisms consuming it locally, and from the outside it would look completely dead. The same enzyme family that lets you take your next breath may have spent 1 billion years keeping oxygen a secret. Every breath you take right now is powered by a molecular machine that has been running, essentially unchanged in its core design, for close to 3 billion years. It is older than any animal, any plant, any fossil you have ever seen. It predates the ozone layer. It predates breathable air. The enzyme evolved first. The atmosphere caught up later. We tend to think of Earth's history as geology shaping biology. Continents move, climates shift, and life adapts. But this research flips that. Biology shaped the planet. Life did not just wait for oxygen to appear and then learn to use it. Life grabbed oxygen the moment it existed, used it for energy, and in doing so, held back the transformation of an entire planet's atmosphere for longer than complex life has existed. And somewhere out there, right now, there might be a planet doing the exact same thing, alive, breathing, invisible. If you want more of the latest science that rewrites what we thought we knew, there is always something new. I will see you in the next one.